Good afternoon to everyone joining from Estonia, but very good morning to Professor Mehta, who is joining us from States today. Thanks for being here so early. And it is my pleasure to introduce our today's guest lecturer, Professor Mehta Kuehn, an Associate Professor of Biochemistry and Molecular Genetics and Microbiology at Duke University in North Carolina. Professor Kuehn earned her PhD in molecular microbiology and microbial pathogenesis from Washington University, St. Louis. And she completed her postdoctoral training in UC Berkeley. For the past 20 years, her research has focused on bacterial extracellular vesicles and their role in infectious diseases. Her lab has developed widely recognized methods to isolate, analyze, and study the vesicles from both gram-negative and gram-positive bacteria, which has led to groundbreaking genetic research in the field. She has also investigated how bacterial extracellular vesicles interact with host cells, how they trigger immune response and toxic responses, as well as their role in antimicrobial activity and their information transfer between cells. Today, Professor Kuehn will share her insight into this fascinating field. Let's give her a warm welcome. Professor Kuehn, we are ready to listen to your talk on mechanistic and functional insights into interkingdom communication by bacterial extracellular vesicles. Now the floor is yours, Professor.
Thank you so much for that introduction and the invitation. It's a pleasure to be here virtually. I wish I were there in person. It'd be lovely to see Estonia. I'm going to see if I can share this properly. Let's see if I can do. Can you see my slides?
Of course, yeah. It's in presentation mode. We can see nicely, yeah.
Yeah? Okay. So, yeah, welcome to a little bit of Duke University. This is what it looks like this morning. It's a beautiful day here. Starting, we're about to have some snow, which is very exciting for us in North Carolina. So I wanted to talk to you today about interkingdom communication. I understand that you have some interest in extracellular vesicles as shuttles of information and EVs in all that are produced by all types of cells. Extracellular vesicles are made by all types of cells and are very good at transmitting all types of molecular information between kingdoms. So I'm happy to answer questions along the way if you get my attention somehow on this thing. It's been a while since I've done a virtual presentation, but I'm happy to clarify or answer questions as we go as well, since this is a small group. So let's see if I can, yeah. So a brief outline of what I thought I'd go over today, introduce you to extracellular vesicles made by bacteria. I don't know if everybody is on the same page, so I'll introduce some concepts there in general. And then I'll talk about several interkingdom interactions that we've been studying over the years that might be of interest to you. And then we'll sum it up at the end. So just to recall, bacteria live everywhere. We often think of them in a health setting as pathogens, but really they exist in our environment. They're in the soil. They interact with animals out in the wild. They're in the environment, and of course also in and around humans. So bacteria are everywhere, and all bacteria secrete vesicles. We have yet to find any species that do not secrete any vesicles. It's hard to prove a null hypothesis in this case, but wherever we've tried to purify vesicles, or others have, we can at least produce them in culture. So, what are these extracellular vesicles produced by bacteria? So, let's see if I can get my laser pointer here. Yeah. So gram-negative bacteria are composed of two membranes, as you probably recall, an outer membrane and an inner membrane that are separated by a periplasmic space in which there's the peptidoglycan, the cell wall of the gram-negative bacteria. And the outer membrane can bulge out and pinch off, producing an outer membrane vesicle. So those are the extracellular vesicles produced by gram-negative organisms. They are consisting of the outer membrane of the bacteria with a luminal content that is composed of periplasmic components. There are also more sophisticated, I guess, examples of vesicles, of extracellular vesicles produced by negatives that also have the inner membrane. So they are doubly membraned vesicles. They're called outer inner membrane vesicles. And so those could contain cytoplasmic content. And depending on the bacteria, those can be more or less commonly seen. Gram-positives also produce vesicles. They only have one membrane, a single limiting membrane, the plasma membrane, which is similar to the inner membrane of gram-negative bacteria. But they are surrounded by a cell wall. It's more challenging to visualize or model how those vesicles might pinch off and be released by a gram-positive because of that thick cell wall, that thick peptidoglycan layer. However, they do make vesicles. You can collect them and analyze them and study them. And they are composed, again, of the membrane, the limiting membrane of the cell, and cytoplasmic content in this case. So these vesicles are considered, we can define them as detached spherical blebs that derive from an intact bacterial envelope. So we're not talking about cellular debris or recircularized membrane from lysed cells, which can happen and might contaminate one's vesicle prep. But we're really interested in my lab in the intact produced vesicles. And this vesicle biogenesis process can be observed. And that's one of the, more powerful images that we can come up with, is the actual production of vesicles by an intact cell. So this was a key observation made in collaboration with Claretta Sullivan, where we could visualize by atomic force microscopy in a kind of time-lapse set of AFM pictures the production of a vesicle from E. coli. So this is done at a low temperature. The stage is at low temperature. The E. coli are live in this case. So these cells are being imaged over time, and you can see the time lapse here going on. And then you can see in this point at this E. coli cell where this bleb is forming, and it's finally resulting in what appears to be the release of this vesicle at this point, and then the intact cell membrane that it remains after this event occurred. So that was very satisfying to see in this AFM experiment, that this was produced by intact cells. We had a lot of biochemical evidence that this was occurring by intact cells, but it's just nice to see it with your own eyes. So the other thing that we noticed is when we inoculate animals with, in this case, enterotoxigenic E. coli, what we saw going into the animals when we did scanning electron microscopy was fairly smooth cells. So most of the cells, about 90% of the cells looked like this going into the animals. So we inoculate ETEC intragastrically, and then after 2 hours, we retrieved those bacteria out of the intestinal tract and plated them and imaged them immediately. And what we saw was that those cells were bumpy. They had a lot of vesicles on their surface. And concomitantly, these cells actually had a reporter in them to indicate that those cells were also stressed. So there was a sigma E reporter plasmid in these cells that turned on when these cells were intragastrically administered into the mouse. So this corroborated with our genetic evidence, which I don't have time to go through today, that the stress response pathway in E. coli not only activates pathways like sigma E, but also vesicle production. And this was really interesting because we know that virulence components are often modulated by their environment, and coming into a host, as shown in this experiment, from a nice LB broth into the gastric environment of a mouse gut, is stressful for the E. coli. And the vesicles that are produced by enterotoxigenic E. coli can carry virulence factors. And so together, these vesicles can be induced by stress, induced upon entry into the host, and in that way transmit potentially virulence factors at that point inside of the host. So there are many factors that promote virulence that are mediated by vesicles that we've studied over the years. I'll show a few examples of those. Toxin transport being one. I'll show you a brief slide about that. Endotoxin is LPS, the outermost lipid of the bacteria. And so because vesicles are composed of the membrane of the bacteria, they are great at transporting endotoxin and transmitting that into a host and causing an inflammatory response. We've also studied how bacteria can use vesicles to remodel their outer membrane, which enables survival in the host. So the vesicles can enable turnover of the membrane components by quickly exiting those vesicles out into the environment and allowing the cell to replenish, if you will, the components of its membrane with a new set of components that are more adapted to function in the host. So we can see that occurs for lipids, for example, that are more adapted to the host environment compared to the previous environment that they were in. So a real quick summary of how we purify vesicles and to study them. We simply grow bacteria in cultures. And that's where a lot of the variability, if you will, comes in. You can grow these in different broths, in different conditions, temperatures, stress conditions, you name it, you can change it, right, in vitro. So you pick your conditions, pick the amount of time that you're growing the culture in, and that's the vesicle. You're going to get the vesicles that have made up to that point in time. So typically, what we like to do is take mid-log growth phase cultures or nearly at the end of exponential phase. That's our preferred time to isolate vesicles from cultures because you have the least problems with any dead cells or debris. And then we pellet the cells out of that culture, take the supernatant and sterile filter it to rid the preparation of any remaining cells. Then take that cell-free culture supernatant and ultracentrifuge it, so 150,000 times G, pellet that, and that becomes what we think of as the crude pellet, the crude extracellular vesicle containing pellet. And then that pellet material is floated in a density gradient. So we've optimized this to be in an OptiPrep solution, which reduces the osmotic stress on the vesicles. The vesicles will float in this density gradient. Their buoyant density will allow them to float up in the gradient over time. We typically do an overnight spin, and then collect fractions from the top of the gradient down, and then analyze those different density fractions for protein content, lipid content, activity, whatever it is that we're looking for. We can see by electron microscopy which fractions contain the vesicles, and they can be visualized by negative staining, and that's seen in this picture. So different vesicle preps, different bacteria will generate vesicles with slightly different densities. So you kind of have to tweak this to be commensurate with whatever bacteria you're working with if you use this type of a technique. All right, and then you can quantify and size the bacterial vesicles. Many ways to do this. We use typically electron microscopy to look at the diameters of the vesicles. You can get heterogeneous composition. In some cases, it depends on the bacteria, the species, the strain. If you have a very heterogeneous population of vesicles or a homogeneous population of vesicles, it's hard to predict what you'll see. We don't know what causes the heterogeneity in size of the vesicles. All we know is what we see. Sometimes it's heterogeneous, sometimes it's homogeneous in size. And we can measure the protein content by Bradford. That's the easy way to sort of generate a dosage. It's just by a protein content. The lipid content is measured using a fluorescent lipophilic dye, which intercalates into the membrane. So it's an indirect method of lipid quantification. And as I said, we can do transmission EM to visualize the vesicles. So what do we see in these vesicles? The bacterial vesicles from gram-negative bacteria will contain, as I said, endotoxin, LPS, because it's the outermost lipid of the outer membrane. They'll contain periplasmic proteins and outer membrane proteins that are embedded in that membrane. But it will be a subset of those envelope components. So not every single protein and every lipid and every periplasmic component is packaged into vesicles. There is a selective process going on. We've been studying that. Again, I don't have time to go into those studies, but understanding the selective composition of these vesicles and the basis for that is really intriguing to me. We are trying to figure out what the rules are for that process, and we know some of them, but not all of them for sure. Gram-positive vesicles contain the plasma membrane proteins, the intermembrane proteins, since they only have the one membrane. Phospholipids are present in both gram-negative and gram-positive derived EVs. Peptidoglycan, at least fragments occur in these vesicles. We see covalent and non-covalently surface-attached proteins, sometimes called a corona now. there's a new word for that that I've learned. Nucleic acids can be externally associated with the vesicle, internal to the vesicle. They can be tightly associated with the vesicle, and we'll see examples of that today. And also small molecules, metabolites, these are present as well. And we're just now starting to understand or even just characterize the metabolome of extracellular vesicles. So they contain a whole myriad of molecular information, but it's a subset. It's a subset of the whole bacterial cell. And you can see that visualized in this SDS page example, where the vesicle fraction differs, in this case from Pseudomonas aeruginosa, from the outer membrane protein profile. So that's just a visual example of a typical prep where we would see some proteins in common between the vesicles and the outer membrane, but also differences, notable differences in enrichment or depletion of components. And again, this characterization, these vesicles, the composition depends on the strain, it depends on the environmental conditions that the strains were grown in. There's a lot of variability that could be observed depending on how these vesicles are prepared. So these vesicles have a unique ability, I guess, to be a secretory pathway for insoluble materials. So typically we think of secretion systems by bacteria as secreting soluble components, but vesicles are a way to secrete insoluble components, and they are a way to secrete a bolus of a complex mixture of compounds that can be addressed to a specific and distant site. So rather than have one toxin that just leaves the cell and intoxicates a host cell, you can have a whole bolus of material, of PAMPs and proteases, et cetera, that all travel together and are targeting a cell at the same time. So you can think of that as a really different and unique sort of way that bacteria can interact with its environment. The membrane provides protection against proteases, nucleases in the environment. And we've seen that in many cases. It's really hard to degrade or these vesicles, even when you're trying on purpose to destroy their structure. It's very hard to do. So our studies have demonstrated that these vesicles are produced and secreted both for virulence-related and basic physiological functions. So we've been studying a number of these. Many groups around the world have also been working on various aspects of the biological functions for bacterial vesicles. So some of them are shown here, and I'll just highlight a few of the studies that we've done. So they can function in phage and antimicrobial as decoys for these components in the environment. They can serve as communication devices. They can serve in virulence and traffic virulence factors between the bacterium and the host, and we'll look at that. Horizontal gene transfers of interest to lots of people. We haven't studied that directly. We have studied more the transfer of RNA and DNA in the aspect of immune modulation. I'll tell you a little bit more about that later. Again, the cargo in the vesicles is protected from degradation by proteases and nucleases. Vesicles can also play a role in biofilm formation and maintenance. I don't have time to talk about that aspect this time around. So let's look at virulence factor delivery for a moment. Like I said, the vesicles are upregulated during virulence associated environmental changes. And at the same time, those vesicles can carry virulence-associated components like toxin. So we studied the enterotoxigenic E. coli heat label enterotoxin called LT for short. And LT is a AB5 toxin, meaning it has an A component and 5 B components. They are assembled in the periplasm of enterotoxigenic E. coli, as shown here. So these 5 B subunits and the A subunit form a toxin, which is then secreted by the type 2 secretion system. And what we found is that this toxin is actually adhesive to the LPS, the lipopolysaccharide lipid that's coating the cell surface. And as a result, vesicles that are produced by ETEC contain the LT toxin inside the lumen because it was in the periplasm, but also bound to the surface of the LPS on the vesicles. So these are LT positive outer membrane vesicles, and they are released by enterotoxigenic E. coli, and they can interact with a mammalian cell very specifically via the receptor for the toxin, which is ganglioside GM1. And the toxin can bind both the vesicle surface and the GM1 to kind of bridge those the vesicle to the mammalian cell. As a result, the mammalian cell, in this case, intestinal epithelial cell, internalizes the vesicle, again, in a specific endocytic process that's guided by the GM1 internalization mechanism. The vesicles are then trafficked intracellularly, and the toxin is released and enters the retrograde transport system sort of backwards through the secretory pathway, through the Golgi, through the endoplasmic reticulum, and into the cytosol, where the A subunit is processed into an active form, and that causes the enterotoxin response that's found in the intestinal epithelium, which results in the water efflux and intestinal diarrhea, which is the problem with the enterotoxigenic E. coli infections. But at the same time, like I said, there's this bolus of information of the other PAMPs that are also entering the cell. So those, the LPS, for example, and the peptidoglycan fragments can activate immune responses as well inside of the cell. So it's not just the enterotoxin response, there's additional inflammatory responses that occur in these cells. And we can visualize this process here by electron microscopy, as you can see, by immunogold labeling and by immunofluorescence of intestinal epithelial cells internalizing fluorescently labeled vesicles. And they really just aggregate. You can see these, the material in the cell just aggregate. It's not degraded per se, because that fluorescence would go away if it was in a acidified component compartment. So this is one example of vesicles trafficking pathogenic components. So now I wanted to go on to some other functions of vesicles, and specifically consider the fact that, as I said, there's not just pathogens out there. There are many non-pathogenic bacteria, and they're making vesicles too. So why would they be making vesicles? There must be something in it for them. It's a big expenditure of molecular energy to make vesicles. And so we considered, how might these vesicles help bacteria survive? Because evolutionarily, there must be some advantage to making these vesicles. And so we considered, in the environment, there's a lot of molecules that are attacking bacteria, namely antibiotics, naturally made antibiotics. There's a fight for bacterial colonization, an environmental niche where nutrients are occurring, where the bacteria can survive. So bacteria are constantly bombarded by antimicrobials. So we considered that maybe vesicles are a way to provide a defense against natural antimicrobials. So we decided to study that using cationic antimicrobial peptides as an example of a antimicrobial that bacteria would see. So in this experiment, what we've done is look at E. coli challenged with polymyxin and a cationic antimicrobial peptide. Increasing concentrations of polymyxin decreases the wild type bacterial survival. Now, at the same time of doing these studies, we were looking at a whole library of mutants of E. coli that may overproduce or underproduce vesicles. And one of these mutants is shown here, YIEM, which overproduces vesicles. So this is a mutant of E. coli, a single gene that hypervesiculates. And you can see that that bacteria actually survives better in the presence of the polymyxin. So that indicated that the production of vesicles was ameliorating survival in the presence of this antagonistic antimicrobial. So we studied that further by just specifically co-incubating vesicles with polymyxin and then adding that to E. coli to see if the vesicles could absorb the antibiotic out of solution, essentially. And what we found was as we increased vesicle content, the ability of the bacteria to survive in the presence of increasing amounts of polymyxin increased. So the addition of vesicles helped bacteria survive higher polymyxin concentrations. They could absorb the antibiotic out of solution. So here's another example of that type of an experiment where we have E. coli in the absence of any of these antibiotics or vesicles. We have 100% survival. When we add polymyxin B, survival is reduced to less than 10%. When we add vesicles to the polymyxin, that survival of the E. coli goes way up, almost to 80%, because in this case, the vesicles are absorbing that polymyxin, allowing the bacteria to survive. Same thing goes for colistin, which kills the bacteria outright. But when the colistin is also incubated with vesicles, the bacteria can survive. So vesicles are a means to protect bacteria from antimicrobial peptides. We studied whether the bacteria are actually producing vesicles in response to these antimicrobials, and we found that to varying degrees, different bacteria do induce vesicle production upon exposure to polymyxin in this case. So ETEC already produces a high number of vesicles. It's a high just baseline producer of vesicles, but polymyxin treatment induced that even higher to, I guess, 6 to 7-fold higher. Pseudomonas aeruginosa also produces a high level of vesicles, and it also could be induced to produce more with polymyxin. Same with Klebsiella pneumonia and Vibrio cholera. So in each of these cases, we could induce vesicle production with polymyxin. So one thing to consider when you can think of EVs out in the environment is, once they're released, the bacteria that produce them sort of is producing them for all neighboring cells also to benefit potentially from them. So do bacteria help each other survive in a hostile environment was the question that we posed. So we had a freezer full of vesicles and bacteria, and we thought we'd just mix and match and see what happens. Do bacteria cross-protect each other from antimicrobial? And the answer was yes and no. So here in this case, the ETEC vesicles were co-incubated with polymyxin and then added to ETEC culture, and they were very good at protecting those ETEC from the polymyxin. And as you go down the diagonal, these are self-protection. So Vibrio cholera vesicles, protecting Vibrio is pretty good. It's better. Klebsiella vesicles are better at protecting Klebsiella. Pseudomonas aeruginosa vesicles are pretty good at protecting Pseudomonas. But what was interesting was the ability, the sort of cross-protection ability. That, for example, Klebsiella pneumonia vesicles were really good at protecting Pseudomonas from the polymyxin attack. So you see examples of this in various places on this graph. We don't know the details of why there's these differences, but it's interesting to as a jumping off point to consider who's protecting who. But we did notice in this case that there's an anomaly here where Pseudomonas aeruginosa vesicles in the presence of polymyxin actually killed Vibrio cholera. So there's an example of the opposite effect where there is an antagonistic effect of the vesicles from one species towards another. And so whether these vesicles somehow potentiate this antibiotic to better interact with Vibrio, or if there's a means by which the vesicles maybe bring the antibiotic closer into the Vibrio so that there's a higher I don't know, concentration of antibiotic at the surface of the Vibrio. We don't know the answer to that, but it's interesting just to consider what's going on in the environment, because the bacteria are not living alone and they are competing for the same nutrient sources out there. And they're defending against similar attacks. So food for thought. So when we think of bacterial populations, there's the OMV producer, the vesicle producer, which can serve as a defensive role or an offensive role, but there is a cost to that, of course, a molecular cost, an energetic cost. So considering the sort of economics of the community is an interesting further road to pursue at some point to consider the economics of the cheaters and the producers and that kind of thing. So are there other threats that bacteria face? Sure, phage. There's 10 times more phage than bacteria on this planet. So phage are constantly attacking bacteria, and maybe we considered that these vesicles can also protect against phage infection, and they can. So E. coli can be attacked by T4 phage. So we looked at plaque forming units. So if there's plaques, then they've been affected by phage. So when we infect E. coli with 10 to the 6 T4 phage, we get 100%, what we called 100% plaques. When we add vesicles, there's no plaques. Vesicles don't cause a phage infection on their own. 10 to the 5th T4 phage down here. So that's the difference between 10 to the 6th T4 phage. This is 10 times fewer. When we incubate these 10 to the 6th T4 phage with vesicles, with E. coli vesicles, we can bring down that titer of the phage infection by tenfold. So these vesicles are very good at protecting E. coli from phage infection. And we looked using a chloroform disruption assay to see if the vesicles irreversibly bind to the phage, and they do, because we added chloroform to various points in time, and we did not see an increase in plaquing. The numbers were very low. So this indicated that the vesicles were irreversibly bound to the phage. And we could even visualize that, which is always satisfying. When you can see it by electron microscopy, here's a T4 phage that's actually bound to the surface of a vesicle. And this is actually hard to capture, this image, because when we first did this experiment, you couldn't even see the vesicles anymore because of the number of phage that were kind of coating the vesicles. So pretty much as many as can actually physically bind to these smaller vesicles. They just cluster the phage and take them out of commission, if you will. Okay, so now I wanted to just touch on some immune modulation and the transfer of nucleic acids into mammalian cells via extracellular vesicles. So in this case, I had a student, Blanca, who was interested in looking at particularly RNA associated with extracellular vesicles. So we moved to study this in Staphylococcus aureus gram-positive, because these package cytoplasmic material, and cytoplasmic material is going to contain presumably some nucleic acids. So she studied the incubation of vesicles with macrophages as sort of a reporter system to indicate whether these vesicles could transmit nucleic acids into macrophages, because the macrophages would respond to nucleic acids from the bacteria by eliciting an interferon beta response. So she incubates the macrophages with MVs, the membrane vesicles from Staph aureus, and at various time points looks for an interferon beta response and other interferons, and found a high response to the vesicles from Staph aureus. So that's indicating that these vesicles are internalized into the macrophages and are carrying bacterial nucleic acids. So then we delved further into exactly what is being transmitted and how it's being transmitted. And specifically, where is this nucleic acid present? So she used, like I said before, these vesicles are really good at protecting nucleic acids and proteins from degradation. So it took a lot of work to actually get this experiment to be helpful using a variety of nucleases. Benzonase turned out to be the most useful nuclease that we found that could digest nucleic acids that were just also present in the extracellular vesicle mixture, and just leave nucleic acids that were internal or potentially also external, but very tightly associated and protected to benzonase degradation. So we added those to cultured macrophages. And what you'll see is that if we had untreated vesicles, this was our interferon beta response here around 175, which was reduced upon benzonase treatment, you can see. So it's a significant reduction, but it's not zero. So there is a lot of nucleic acid that's generating this interferon beta response that is protected from digestion by nucleases. But some of it is external. So we have, again, a duality of where this material exists. So there's highly protected nucleic acid that induces this potent interferon beta response in macrophages. Then we studied the internalization process and used dinosaur as a mechanism to inhibit dynamin. This inhibitor prevents the internalization aspect of the endocytic process. And you can see that the interferon beta response was greatly diminished when we added dinosaur. And we could visualize this internal localization of the MVs using a cytoRNA select staining in green. So when that co-localizes with the early endosomal marker, EEA1, you will see yellow staining. You can see some yellow staining in these cells. We further looked at the endocytic process, looking at exactly which TLRs are activated, and looking at whether bacillomycin or chloroquine could inhibit this process, because that would show that there was acidification that was needed, and that there is maturation of the endosome that's needed, that's contributing significantly to the vesicle-mediated interferon beta response. So bacillomycin and chloroquine inhibited a great deal of the interferon response. And these were all done with the benzonase treated vesicles. So we're looking at only the response to the highly protected nucleic acid in these vesicles. So then we went down the line of various TLRs that could be involved in this immune response, using macrophages with specific deletions in TLRs, TLR3, 7, and 9. And each of them significantly reduced the response, but it was not a zero response. There wasn't a single TLR that was responsible for the entire interferon beta response. So that told us that there was a combination of nucleic acid types in these vesicles. There's double-stranded RNA, tRNA, single-stranded RNA, as well as CpG DNA that was present in these vesicles, because each of the TLRs was important to add in an additive way towards that interferon beta response. Okay, so finally, I just wanted to end with this vignette of thinking a little bit more broadly about vesicles in the world and the environment, and their ability to communicate externally. And so this is a study that Hannah McMillan did in the lab. She was interested in plant and bacterial interactions in the environment. And so she chose to study Pseudomonas syringae and Pseudomonas fluorescens, so considered a plant pathogen and a commensal, if you will. It's often used in engineering of in crop promoting health of crops, this commensal fluorescence. So she isolated vesicles from these cells in complete media as well as minimal media. Minimal media, in this case, enhances type 3 secretion system in these in the pathogen in PST and the syringae pathogen. Syringae because it has a type 3 secretion system syringe that is known to contribute to the pathogenesis of this organism. So she wanted to look at vesicles made in sort of robust conditions where nutrients were freely available, and those under conditions of sort of minimal nutrients, and see if they had different functionalities in interaction with the host cells. So this is just an analysis of the types, the characterized isolated vesicles. And essentially, there's not a difference in protein content or size, lipid content, number of OMVs. Everything was very similar. But then we used these vesicles to infect plants. And normally, plants that are challenged with Pseudomonas syringae cause yellowing. And that's seen in this upper panel. So green leaf of Arabidopsis treated with Pseudomonas syringae bacteria turns leaf yellow. When we pre-treated those leaves with vesicles, the leaf yellowing decreased. Both the vesicles from the syringae, which is the challenge organism, but also the fluorescence vesicles caused the leaves to resist this yellowing by the PST. And we could analyze that numerically to figure out what the dosage was of the vesicles that allowed this protection of these leaves. And notably, the PST vesicles were very good at this, but the Pseudomonas fluorescens vesicles were even more potent. So we had responses much in concentrations much lower than the PST vesicles. So PF vesicles are very potent, very good at protecting plants. And also notable, we tried different bacterial vesicles. We thought, well, if PF vesicles are so good at this, maybe other gram-negative bacteria vesicles can do this protection as well. So here's PST protection. Here's Pseudomonas aeruginosa vesicles. They can protect a little bit, not much. E-tech vesicles, they can. This is enterotoxigenic E. coli. They can protect these plants against PST vesicles. Staphylococcus aureus, those vesicles that Blanca used, were not very good at protecting. This was just, like I said before, if you can track the functionality of different fractions of vesicle prep to see if it tracks with the vesicle fractions of the secreted components, and it does. So the proteins and lipids peaked here in fractions 5 and through 7, and you can see that the most protection offered to these plants was in these fractions 5 through 7. So it was commensurate with the vesicle peak. And then notably for any plant aficionados out there, type 3 effectors are like the thing for virulence characteristics of plant pathogen, like Pseudomonas syringae. It makes this syringe in which it secretes via the type 3 secretion system virulence factors directly into the plant cell. So we thought, well, is this mediated somehow by this type 3 secretion system? And what we found is no, the vesicles pre-treated with the plants pre-treated with vesicles from type 3 secretion system mutants were no different from plants treated with the wild type vesicle preparation. So type 3 secretion system is not involved in the vesicle-mediated protection. All right. And then finally, we also looked at the stability of these vesicles. Again, they're highly nuclease resistant. We talked about that before in the other section, but they're also really resistant to physical parameters like sonication, boiling, adding protease. So this protective factor is very, very stable. That's mediating this protection in the plants. And we looked at oocyte, oomycete pathogens like Hyloperoptera Arabidopsis infection of Arabidopsis. Vesicles are also able to protect against oomycete pathogens in Arabidopsis as well as in tomato plants. So there's a lot of cross-kingdom transport process going on here, molecules that are able to activate a protective immune response from one species protecting against another species. There's some fascinating interkingdom communication going on here. So finally, so hopefully you got some idea about virulence, non-virulence associated functions of bacterial vesicles. Vesicles produced by gram-negative bacteria, outer membrane vesicles, can absorb antimicrobial peptides. These can act to reduce or potentiate antimicrobial activity in polymicrobial communities, considering the environment in which they're occurring. This can be very complicated if they're acting for the good or for bad. They can irreversibly adsorb bacteriophages in the environment and prevent cell infection. They can elicit immune responses in mammalian cells. They can elicit immune responses in plant cells. So they can conduct interkingdom communication. And exactly which components of the vesicles are promoting this offense or defensive traits are that's of interest. Is it is it the same component? Is it different vesicles? Is it the same for different hosts? What are these components? Is it lipid, protein, toxin, nucleic acid? There's many choices. Is it a combination effect? So this remains to be studied. Further food for thought. And so finally, I acknowledge a great group of people who have done this work on the slide. We see Blanca and Hannah, who've done the work I talked about, and the collaborators that were involved in this project, I appreciate, and the funding. So I'll take questions if you have any.
Thank you very much, Professor Mehta, for the clear and very interesting presentation. I'm certain that our audience will have very interesting questions for you. Actually, yeah, one, I think Germany has a question. I can go on.
Yeah, Domin.
Yeah, thank you very much for the very comprehensive and interesting talk. And there's few technical questions I want to ask, and also some observation factors. So the first one is in the isolation, OMV isolation procedure. You have used 0.45 micrometer filter. But when I was practically trying to do that, and when I go through the literature, I felt like it's better to use 0.22 micrometer filter. So, but in my case, I got very little number of OMVs. Maybe that might be the reason. Maybe I would like to get your opinion regarding that.
Yeah, we've done both. I would agree that using 0.22 will probably reduce the amount that you get, and just based on clogging of the filter, if nothing else, it just they can really aggregate on there. And so if you can, it's some vesicles are smaller, some are larger, depends on the strain that you've got. And so you do have to play with that.
Yeah.
There's also the bacterial size, which is a problem. In some cases, if you don't use a 0.22, you get too much contamination. And
Yes.
so it's a tricky business for sure.
Yeah, so
Yeah,
in your case, actually, when you use the gram negative, especially like E. coli and the Pseudomonas, I felt like it's okay to use 0.45.
exactly.
But
Yeah.
and it is for the gram positives, like really teeny ones, then yeah.
Yeah.
I had that thing, and then I was working on the 0.22, and then when I saw your thing, I was like, oh, maybe this is where I did the crap.
You might be losing it there. Yeah.
Yeah, and the other one is like I've seen that most of your work is towards gram negative bacteria, E. coli, and the E. coli Pseudomonas and stuff. But did you see the same observations for the gram-positive bacteria, especially the increased outer membrane vesicle production with the stress conditions?
I'm trying to think of if we did that specific experiment with staph. We were looking a little bit at different media conditions for the Staphylococcus, but I can't think of any specific
Okay.
that we use to induce them.
Mhm. And the last question. Sorry. The thermal stability, the last part that you were talking about, the boiling and all. So is it only observed in MV is isolated from E-tag, or is it with the other bacteria types, or just the Pseudomonas? As I always felt Pseudomonas, it is bit worrisome and bit nasty working with that.
You mean for the stability of this component?
Yes, yes.
So in this case, what we were looking for was the protective component that was carried via vesicles. Sorry if I went over that too quickly. Whatever the protective component is, that it's still active even when we boil this preparation.
Okay.
Whether that's also the case for any of the other vesicles protective effect in other scenarios, I don't know.
Okay.
Yeah. Typically, boiling would inactivate it, like a protein toxin. So, yeah. But in this case, it would seem that whatever component is protecting these leaves from infection is very stable to boiling, which was interesting. It leads us to think it's a metabolite or a lipid component, potentially.
Yeah, thank you very much.
Sure.
Okay, thank you, Durmini. Next, we'll give the opportunity to Alireza, is the PI of our lab. We can't hear you. I think you are in mute. You are mute, Alireza. We cannot hear you. You are mute, yeah.
Okay, I think, can you hear me now?
Yes,
Yes.
now we can hear you.
Yeah, sorry, apologies. I thought Suranga was before me, but you gave it to me. But okay.
Yeah, sorry, I didn't see who hands up first, so it was my mistake.
Okay.
I
Sorry.
apologize.
Okay.
Yeah.
But apologies, Suranga. But thank you very much. It was really interesting, and considering that you mainly work with the mammalian ones, now we have got also the plant and algae, but bacteria, the amount of the work and also the things that you guys have progressed has been phenomenal in the whole EV life. One question I have is a general one. Do you think that the EVs that are produced by bacteria, for example, different kind of bacteria, would they have specific targets? Is there a specificity there? Is it a trophism there? Do you think that is the case, or you think that no, it's just any kind of EV for any kind of target or any kind of interspecies communication also can be used? Or what is your feeling, or do you have any definite answer for that?
Right. Yeah, we think about that a lot, the kind of the purposefulness of this secretions mechanism, right? So in the case of some kind of functionality, like phage infection, I mean, the vesicles harbor the same receptor as the cells in this case. Are the vesicles releasing that receptor on purpose, specifically for this anti-phage defense system? I don't know if that's the case here. What I didn't show here, but we've done the study on outer membrane lipid remodeling, where we see changes in the, I don't know where to show this best, maybe like here. The changes in the outer membrane lipid, right? As let's say cells enter macrophages, they enter a acidified compartment, a low pH, low magnesium concentration. Bacteria have to change their membrane in order to survive inside the macrophage endosome. And the vesicles that are released have a specific composition that does not reflect the bacterial composition in that case. So in that scenario, there is a directed composition of enrichment. So, and the same is true when we look at the vesicles released by ETEC. My slides are going funny. These vesicles that are released by ETEC, and they are enriched in the LT, the toxin, which is directing them into epithelial cells specifically. It's a very specific molecular receptor interaction. So in that case, there is directed transport. Yes. Yeah.
Maybe not.
So
yeah. Okay. Then the next one, just this question, you in mammalian cells, in mammalian EV, we know that not all the vesicles have a cargo of nucleic acid. Even when we are speaking about microRNA, the other cargo that is in there, there is quite a lot of evidence. Sometimes we think maybe even only 10% of them have a cargo. And there is nowadays, especially, a lot of debate is coming that do at all that cargo of nucleic acid has any functionality, or is it even, there are people saying that a liposome would be a better carrier of the nucleic acid compared to the EV. You very nicely showed that work with TLRs and how it was affecting the nucleic acid was, if I understood it correctly, was affecting it. What do you think? Do you think that it is I know the protein cargo, I can imagine the toxins, all of that. But when it comes to nucleic acid, what do you think? Do you think, is it really cargo, a nucleic acid cargo that is transferring the function of the bacterial EVs?
Right. So this is a question grappled with for a couple of decades. There is definitely evidence published that shows that these vesicles can be what is termed transformosomes or something like that, right? They're able to transmit a plasmid from one cell to the other.
Yeah.
Okay,
Yeah.
so it's published, and we were interested in this, of course, to see, is this a means by which cells, bacteria can actually transmit genetic information, important, the entire, an antibiotic resistance gene, obviously important, right? So to me, that would have to involve a specific mechanism of release and uptake. And so we thought we would look at it and tried to recreate what had been published. And I can say that if you take a cell with antibiotic resistance plasmid and isolate vesicles from that, and then treat it with DNase, to digest anything extracellularly that might have burst out, and then incubate that with a recipient naive cell that is not resistant, and co-incubate that for some amount of time in some conditions, you can get transformants that contain that antibiotic resistant plasmid, right? But the frequency that we saw that is not high. It's not something I would base an evolutionary method of transmitting nucleic acid from one cell to a recipient cell. It didn't seem in our hands to be very efficient. Does it need to be a valid system of transferring nucleic acid? I don't know. Are the conditions that we used in the lab suboptimal? Probably. we could never get our hands on exactly, is it magnesium? If you add EDTA, if you do different temperatures, time, whatever, all these different parameters, we couldn't figure out a mechanistic means by which this transfer occurred. So that's where that kind of stopped for me in thinking about this as a means to transmit true gene information. Now, that's why when we studied the nucleic acid transfer, interkingdom transfer, if you will, we used Staph aureus and a macrophage response to nucleic acids in general, as a PAMP, not as a gene. So yeah, this response is to the molecule, right? The interferon response is to the nucleic acid molecule rather than a gene specific effect of a silencing or microRNA,
Yeah,
process.
exactly. Genomic or
Yeah. So I also think, in my estimation, it would take so much vesicle mediated transport to make an impact in a host at a gene silencing level that it would not be physiologically feasible.
No, thank you very much. That was and thank you very much again for your very, very comprehensive. And sometimes when I was looking at your slides, I was thinking I have to pack everything, go away. You have answered all the questions. I don't need any more.
Thank you. Thank you.
Ranga, please.
Yeah, I feel.
Yes, Ranga, please.
Yeah, thank you very much, Dr. Mehta. It was very, very interesting. I have actually a couple of questions. I'll be asking very quick questions actually. So one of the things actually hit my mind when I look at your gram negative and gram positive, especially packaging wise. This is actually something extending Aliresa's one of the questions on nucleic acid packaging. So actually, we were thinking of using fecal EVs to look into the detail of microbiome, the gut microbiome. So would you think it's a good good idea? It's something a good idea to use fecal EVs, because we thought that it could be a good source of nucleic acid from the bacteria in the gut. So I have seen many papers even, but still some people are challenging, saying some organism they never ever produce any EVs with the nucleic acid, specifically DNA. So some people are saying, okay, you should use RNA in your phylogenetic analysis rather than DNA. So what do you think? Do you see any difference between
I
DNA?
haven't seen a selectivity to DNA versus RNA. In my estimation, from our experience, it matters so much on how you're trying to detect it. So the means by which people are detecting using prep kits or sequencing it really changes what you see as far as the vesicle associated nucleic acid. We tried all these different methods and we see different sizes of like RNA. So I think it's more in our hands that shows this variability, and I'm not convinced that it's on the bacterial side, that it's like one is more packaging one type over the other type of nucleic acid. I think it's more about our ability to detect it. Because again, when we look at, we use this indirect assay essentially to tell us what the vesicles are containing using these macrophages. I think the mammalian cells are much better at detecting nucleic acids than our methodology in vitro. So your question about using microbiome-derived EVs. So I go back and forth on this sense of we show it here. The corona, if you will, of nucleic acids that are coating these vesicles is not insignificant, right? Even though we think it's debris or something that they've captured that is maybe not of their own origin, even from the bacteria that produce the vesicles, that material can be very tightly associated and can be co-transported. So that's not nothing. That's a valid mechanism of nucleic acid transport, even though it's not necessarily self-derived. So when you talk about a microbiome-derived vesicle, they can be coated with all kinds of stuff from the other microbes that are around, which is intriguing. It doesn't make a simple question of what you're, I guess you need to consider, like, what are you asking about when you do that type of experiment? You isolate them from this kind of dirty mess, right? It's like, well, they're going to be coated with dirt, you know, because they're charged and they're going to pick up charged molecules on their surface. So okay. Good luck. I've seen it done.
Quick question on this. So you have shown some nice data on AMR, involvement of EVs in AMR, like they just protect each other. So have you done anything by inhibiting the secretory pathway and then adding this like antibiotic and see like whether this really is due to the EV? So just only one like it's something non-significant or something, like if you just stop the secretion of EVs and then add the antimicrobials, do you have any idea whether it will turn it back, like so it will become susceptible for the antimicrobials?
I'm trying to think about what you're asking. We can't stop vesicle production, if that's part of your question.
Yes, exactly. That's one part of my question as well. Can you stop it? Like, can you inhibit it greatly? Like,
can you inhibit vesicle production? That would be that's the golden ticket there. Genetically, yes, but an inhibitor to add it, we have not yet seen this type of inhibitor. I think of, like, what would that inhibitor be? It'd be like glue, you know, if you just
like
coat
a mixture, I would say, like a mixture, not a single one, especially like because there are different pathways that are involved, like so there's a mixture of
we don't have that.
And the last question, quick question. So what do you think about using the bacterial vesicle as vaccine platforms? Because Peter was here, I think he has already left the floor, because Peter is, I know he's very much interested on using EVs, bacterial EVs as vaccine platforms to send the antigens.
Yeah. Well, they're great. They're absolutely fabulous. They're great at presenting surface antigens, right? And what better, you know, protective antigen than a surface protein? So it's the right platform for it. They're not living, so you don't have to worry about you know, infection or, you know, not an avirulent cell type or anything. You can hyper express a epitope. So we've expressed heterologous epitopes from Yersinia onto E. coli vesicles, for example, as long as it's compatible and it gets to the membrane, it's great for that. To me, when I started looking at this, again, many years ago, considering this, I thought, who would ever want to use a outer membrane vesicle that's filled with endotoxin? and use it direct as a vaccine, because that's such a response that we're trying to eliminate when we're trying to develop vaccines, is that inflammatory response that we see, like with the HIV vaccine. It's so immunostimulatory, it hurts and it causes bad reactions. So I've considered, looking at, and you can make vesicles from a strain that's less toxic, less endotoxic. It has fewer lipidated LPS. They don't vesiculate as well, but you can do it. But then I thought, why do that when you can just kind of take your epitope and your liposomes and make something that's much more homogeneous and more likely to go through a approval process than this heterogeneous mixture that's hard to get. Why would one want to make vesicles when you could take a cell membrane and just reconstitute a vesicle out of a cell rather than make these natural vesicles? So this is sort of a dialogue I have inside of my head for many years of like, what's the advantage of using a natural vesicle? And there might be some, advantages, but I see a lot of disadvantages that I can't see really working out at a production level scale. That's a lot of work for, I think, something you could get in a different way, but maybe I'm too pessimistic. I don't know.
Thank you.
I mean,
Thank you very
obviously, a lot of they're great at producing protective immune response. So, yeah, they work, but I think there's downsides.
Okay. Thank you. Thank you so much. Thank you so much.
I also have one last question, Professor. I was thinking it was very interesting to listen that this bacterial leaves, they bind to natural antimicrobials, kind of like absorbs them, including the phage. And do you think that the bacteria has the ability to, self eat these bacterial vesicles and kind of change their genotype to be more resistant to these antimicrobials? So, can it be for example, have you done such experiment that you treat these bacterial extracellular vesicles with antimicrobials and trying to put them on bacteria and see whether their genetic phenotype changes or something like that?
So I think you're talking about sort of these experiments. Is that right?
Yeah, OMVs with polymyxin antimicrobial, and then if you put them back on the bacteria, survival, then okay, it's a mechanism then, right? And how what is like the possible mechanism that the bacteria take up these vesicles, because they cannot endocytose them, right? How do they take in them?
Right, they cannot. So that's what makes this experiment really interesting. Because this mixture of vesicles plus polymyxin is being co-incubated with these cells, and there's not endocytosis, as you say, going on. But in this case, where there's this killing effect, like what is happening here is intriguing, because you have this complex of vesicle, Pseudomonas vesicle with polymyxin that's interacting with a Vibrio surface, but it's killing the Vibrio, but it's not because it's getting internalized, right? Because it's not an internalization of the entire vesicle. So yeah, something is going on here. Don't know what's going on. We tried a couple of experiments to see if we could unravel this, but I don't know. I think it's just presenting the polymyxin in a more active form, if you will. And there's something about Pseudomonas vesicles.
So you
or-
think most of the interactions happen on the bacterial surface, not that these vesicles get into
Yes,
the bacteria? Okay,
that's right. Yes,
great.
yes,
Yeah.
yes.
Mhm.
Yeah.
Okay, I know that time has passed, I know, but there's one more question. But if you are, if you're busy and want to leave, we can finish. There's one more question on the text. Can I read it?
Sorry, yeah.
So it says how E. coli is protected by OMBs from phage infection. So the question is by GetNet.
How?
And how
Mhm.
E. coli is protected from OMVs from phage infection, what is being transferred, and how do OMVs reduce T4 phage infectivity? So he's kind of asking what are the key players and mechanism of action involved in irreversible binding OMVs.
So yeah, for the phage infection, the vesicles are able to carry the receptor for the phage, and so the phage interact with the same receptor that they would act on the cell, and the phage are locked in. There's two modes of binding for T4 phage. There's a sort of a reversible process where kind of the phage are checking out if it's the right host. And then there's the irreversible sort of bound form in which the DNA gets injected into the cell normally. But in the case of the vesicle, it's kind of a decoy. It's a mock cell. And so the phage are in that second state of irreversible binding. And so when you take that complex mixture of phage and vesicles and you add it to cells, those phage cannot be released and infect the cells. So that's how the vesicles are able to bind to the phage. And of course, each vesicle has many of those receptors on them, so they can bind as many phage as physically are able to be on that spherical surface. So one vesicle can take out probably, I don't know, 15 phage or so. You can see it by EM. I don't have the picture here, but a whole cluster. You can barely see the vesicle in the center of this cluster of phage. So that's what we see. And of course, those are then floating around in the environment too. And so we considered what they were doing in the environment, in a polymicrobial environment. And they have some interesting effects on other cell types. So they're not inert at that point. They do interact with other bacterial cells and cause downstream effects in other cells. So for example, the E. coli T4 phage cause and effect in Pseudomonas that is unexpected. They elicit a response, probably, again, based on these kind of surface interactions. It's not endocytosis, it's the same situation, but there's a lot of surface-to-surface contact, I think, that's going on, and it's causing interesting crosstalk.
Hope GetNet got the answers. So yeah, thank you very much, Professor Mehta. You spent like 1.5 hours with us today and we learned a lot. And we hope in this year we would hear more interesting and amazing findings from your lab.
Thank you so
best
much.
wishes. See you again sometime.
Okay.
Yeah. Bye-bye.
Bye. Thank you.
Thank you.